Good afternoon, everyone. Hello, 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 hello. We're going to get started. One of the things that I want to do before we move on, finish up malaria, is I just wanted to finish up with one thing that we didn't talk about with EBV or infectious mononucleosis. So we talk about when you can be infectious. So with infectious mononucleosis, you can be infectious while you're in the incubation period of the disease. You don't know that you have the disease because you might have a sore throat, or you might be in the prodromal stage of the disease where you're starting to have a sore throat, but you don't know what is causing the sore throat. It could be strep, it could be all sorts of things, right? So we're going to learn that pharyngitis is a common symptom of a number of diseases. However, once you get into the typical signs and symptoms, once we find heterophile antibodies, we can diagnose you as having infectious mononucleosis. Generally, you only have infectious mononucleosis, the signs and symptoms, once. There are other things that cause similar signs and symptoms to infectious mononucleosis. We'll talk about another EBV disease in just a little bit, or another herpes virus family member in a little while. But really it comes down to that you generally only get it once. However, it can reactivate throughout the rest of your life. So when you get reactivation, it means that there'll be productive viral infection randomly throughout your life. When you're stressed, when you're tired, when you're not eating correctly, when you've driven 38 hours to Phoenix to watch a basketball game, okay? So, and then 38 hours back from Phoenix after partying for 2 days straight. So all of that can lead to symptoms where you start shedding virus again. And then the other thing that happens is that this is one of the viruses, this is one of the diseases that you can shed virus through the recuperation period, up to 18 months after you had the initial infection. So you will be infectious during all of that time. Okay? Does that make sense? So once again, it's different in different age groups. Generally, we only see infectious mononucleosis in people about your age. The disease, if people get it, if children get it when they're younger, they generally don't show signs and symptoms. And where we see the most problems with people having sequelae or something like Burkitt's lymphoma of the jaw is when those children have immune deficiencies. They're malnourished or they're co-infected with malaria. So we've just talked about malaria now. So you guys now know about malaria. So in malaria, this is a protozoan, and this protozoan, again, has a typical life cycle. One of the questions, people are having some problems with a couple of the questions on Connect where it's hard to understand what the answer is that they're looking for. If you have questions, post them. I'll have a Connect. If you guys send me a specific question that you don't think is correct, I will review it and look over it, and if we have to, I'll give credit for it. So there'll probably be some adjustment of scores as time goes on with Connect. The other thing that we're going to talk about with EBVs is there's protozoan, and this is the one place where when I tell you that most antibiotics don't work on eukaryotic organisms, for some reason there's one antibiotic that does work on one stage, like the merozoite stage. Why? We don't know why. Because it's an antibiotic that inhibits, I think, DNA replication. For some reason, it might be able to inhibit the DNA replication of that particular stage of replication, of reproduction. But we don't know why. So it doesn't make any sense that it works there, it just does. We also are going to talk about, we have anti-protozoan diseases or treatments, we have anti-protozoan drugs that we can give you, things like chloroquine, quinine, I think is also one. But the organism is becoming resistant to those treatments. So the other thing that we're going to be seeing throughout the rest of this class is as we go on in this class, organisms become more and more and more resistant to all of the treatments that we have. Now, one of the best things that's happening is we actually had a speaker come and talk to us, and one of our labs is actually working on malarial treatments. And there is real hope for a vaccine, possibly in the near future. We're working on it. There's good feedback on it, but there's no vaccine yet. So when we look at this, you guys need to know the life cycle of the organism. It infects a biological vector. You have to know the organism. You have to know that it is a specific genus, the specific Anopheles mosquito is the biological vector. The Aedes aegypti is the biological vector for yellow fever. Okay? So we have those. It's going to infect the liver first. It's going to go from the infectious stage. Once the mosquito takes the blood meal, it will then migrate through your bloodstream to the liver where it enters intracellularly, reproduces, exits from the liver into the red blood cells, where it remains in the red blood cells for the rest of this person unless they can eliminate it. Some people can get over malaria. Generally, it takes treatment or drugs to help you get rid of it. This organism makes your blood very sticky. It causes your heart to have to pump very hard. And it's especially traumatic or especially lethal in children where their hearts aren't as strong as ours. So when we look at treatment, we're going to talk about anti-protozoan, anti-eukaryotic drugs. But again, one of the other things that works really, really well is we know that the mosquito feeds at night. So if the mosquito feeds at night, one of the best and simplest ways of preventing malarial infections is to simply sleep underneath a bed net. And this bed net has been imbued with anti-insecticidal components to kill the mosquitoes so they can't get through. And so if the mosquito can't bite you, you won't get malaria. It's not transmitted from person to person. It's only transmitted through the malarial, the biological vector. Okay? So one of the best, somebody sent me in my honor for Christmas. I always tell people this is a good, if there's somebody that has everything, it's easy to go into this. There's Nothing But Nets. It's a World Health Organization affiliation where you can go in and buy nets for 10 nets for I think $10, something like that. Where you can go in and help people not get malaria and do it in somebody's name. You know, I'm not asking you to do it for me. Do it for your uncle who drives the Tesla and you can't think of anything else you could get him besides, a signed baseball from whoever. Okay? So anyway. So this is something that we can do. This is something simple. And it makes sense that we can prevent disease by preventing being bitten. Okay? Any questions about malaria? So again, we have real hope that there's going to be a vaccine in the near future. And we have real hope that it might be us, UNC, that provides that vaccine. So who knows? So now we're going to talk about diseases of the respiratory system. And there are a large number of complicated diseases that we're going to get to in the next 2 lectures. It's going to take 2 lectures to get through all of these. Now I want you to remember that we're going to talk about the specifics. Most of the information that I'm going to hold you responsible for is not just on your outline, but it will be on these charts. So print these charts out or print any of the extra material, like that EBV diagram of how EBV infects. It infects the throat, drains through the lymphatics, gets into the lymph nodes, infects blood cells, B cells. You have a productive infection, you have a non-productive infection. I always ask about a productive versus a non-productive infection. Which one makes heterophile antibodies? Which one needs a T cytotoxic response? Which can be detected by a T cytotoxic response? How do T cytotoxic cells find infected B cells? What do they bind to? What do they recognize? All of that is important, okay? Because it all makes sense because we just cleared up how immunology works. So now we're talking about this. We're talking about diseases of the book. Divides these into upper respiratory and lower respiratory diseases. So the first diseases that we're going to talk about are the upper respiratory ones, and then we'll migrate into the lower respiratory ones. And this is just if you don't remember from anatomy or physiology what the organization of the upper respiratory tract and the lower respiratory tract looks like. This is what it looks like. We're going to talk about these as we go on. So the first organism that we're going to talk about is something called Strep pyogenes, and you guys know Strep pyogenes from lab. Strep pyogenes is a streptococcus, means it's gram-positive in chains. Now, there is a whole number of species or specific epithets of strep. There are a large number of these gram-positive chain cocci. So, as a matter of fact, when we talk about this, we're going to talk about how we identify a different strep from each other. One of the things that is important about strep pyogenes is that it has a huge number of virulence factors. You guys already know one virulence factor. This is the organism that has M proteins. M proteins are responsible for destroying C3b. Now remember, in the book it talks about how complement only works on gram-negatives. Well, if complement only works on gram-negative, why does this organism have 3 different mechanisms of inhibiting complement? So it has M proteins, which inhibit C3b, or opsonization. It also has protein G, and protein G is the Fc receptor that binds to IgG antibodies that can't activate complement, and it inhibits opsonization, both things, right? So it inhibits protein G, so you guys know what protein G is. You guys know what the M protein is. This is also the organism that has the C5a peptidase, so it's going to inhibit C5a complement proteins from allowing PMS to migrate to the site of infection. All of that acts on complement proteins. So now you guys all know and understand why those things are important. This has been a very dread disease up until we got discovered antibiotics. If anybody's ever read the book The Velveteen Rabbit and the little boy who almost dies, he almost dies from a strep sore throat. And they have to burn all of his toys because there could be organism, because strep is spread on the surface of toys and on the surface of things. So that was all before, that was back in the day before we had antibiotics, right? So this was a dread disease, and these virulence factors are what allowed it to be a dread disease. So when we have that, we have C5a peptidase. We also have a hyaluronic acid capsule. So this is different from most capsules that bacteria make. Most capsules make some sort of a bacterial sugar coating that makes them slippery. This is a human protein, or it looks like our hyaluronic acid, so it's a cloaking device. So what it does is it wants to cloak strep pyogenes from being recognized by the immune system. We're not going to make antibodies to our own hyaluronic acid. So we won't have neutralizing antibodies to the capsule because it looks like so. These are all good questions I could ask on an exam. Okay? Why won't strep pyogenes have a neutralizing antibody to the capsule? Why does it make why does it make what protein G is? What all of this works as? And notice that the hyaluronic acid capsule masks the peptidoglycan, and also there's something inside called group A carbohydrates. So, because we have so many classes, so many kinds of streptococci, they're impossible to differentiate just based on gram staining, because they all gram stain positive, they're all cocci, they all look like strep. So Barbara Langsfield, when she was looking at these different organisms, she found that every strain, every species of strep has a different carbohydrate there. Because they have a different carbohydrate, she started identifying these into groups that could be identified. She started with group A, and then the next one that she identified was group B strep. And then she went to group C strep. And then she went to group D strep. This is number 1. This is group A strep. And as a matter of fact, in a lot of places, in a lot of literature, in a lot of papers, in a lot of different places, they call this, and I will also use the term gas on an exam, that you need to know what gas stands for. Gas stands for group A strep. This also identifies strep pyogenes. This is specific for the species strep pyogenes. We're going to talk about group B strep. We're going to talk about, we're only going to talk about 2 kinds of strep. Mm-hmm.
What did you just say was specific to group A strep?
So group A strep is that specific A carbohydrate. So it's group A carbohydrate that makes this group A strep. Uh-huh.
So are there multiple Streptococcus bacteria that fall into group A?
Are there multiple?
Are there multiple types of strep that fall into group A?
No. Strep pyogenes is the only group A strep. Okay? So strep pyogenes is it for group A strep. And you guys know it's beta hemolytic, all of that stuff. You're going to be able to identify this. You can identify this in line because you've seen this, right? So this is the kind of organism it is. And these are they're also other, they have streptolysins, they have tissue degrading enzymes. I'm not going to hold you responsible for anything that's not not on your outline. I know that there's a lot of material there. One of the diseases, and because different kinds of, now what is different between strep pyogenes are the number of virulence factors that they have. We're going to talk about some of these, the ones that are on your outline, all strep have. Okay, so all strep has a hyaluronic acid capsule. All strep makes M proteins. As a matter of fact, the other thing that you need to know about those M proteins is that what differentiates strains of strep from each other are those M proteins. So on your notes and in your outlines, it says that we have about 80 different strains of strep pyogeny M proteins based on their M proteins. Okay? So serovars and strains are the same word. Okay? So we will have all of those strains will have a hyaluronic acid capsule. All of those strains will have group A carbohydrates. All of those strains will have G protein. All of those strains will have protein F. All of those strains will have one flavor of M protein. One of 80. Okay? Now, the other things that are there are something called tissue-degrading enzymes, and this is the organism that, if it has a tissue-degrading enzyme, if you've ever heard of necrotizing fasciitis or flesh-eating strep, this is the organism that makes it. This is the organism that can eat your arm off if it has that toxin. But that is so rare, it makes, when it happens and when it occurs, it makes news around the world when somebody has their arm eaten off. Okay, so we're not going to talk about that. I'm only going to ask you the ones that are on your outlines, the one that we see every day in garden variety strep. Okay? So let's talk about garden variety strep. When we talk about garden variety strep, again, they have this protein G that holds the FC region. It can't opsonize and it can't activate the classical complement pathway because it's binding the FC region of IgG antibodies. So, what else do we know? The only thing that strep pyogenes does is it causes pharyngitis. And you're like, well, that's not very bad. It's just a sore throat. It is just a sore throat. As a matter of fact, if that's the only thing it did, we wouldn't be worried about strep pyogenes at all, other than the fact that it causes a sore throat. The other thing that's very important about this is it causes a sore throat, a red throat with pus and tiny hemorrhages at the back of the, we're going to talk about all of these, I'm just reading this off of this chart, tenderness on the lymph nodes, and frequently, less frequently, sometimes you'll have abscess formation. Notice that there's no fever there. There's also no runny, there might be a little fever sometimes, but most of the time, the other thing that with a strep sore throat, and the way that you can differentiate this from a cold, is that there's no runny eyes, no nose, mucus discharge. You just have a sore throat. Okay? So you have this sore throat, and then what happens is the problem that happens isn't with the sore throat. The problem that happens is with the sequelae to this infection. Now, we talked about sequelae before, but when we talk about sequelae, we're going to, we're going to talk about what sequelae are. And when we talk about what sequelae are, sequelae is a condition that arises due to this previous disease or injury. So when we talk about sequelae, so the sore throat isn't the problem. It's our immune response to the sore throat. It's immune response to this organism, and it's based on HLA identity. Not everybody will have this, but certain MHC molecules are responsible for presenting and processing and activating these sequelae. So when we talk about these sequelae, on the left, we now go through the whole infection pathway. We talk about strep pyogenes entered by inhalation, so somebody coughs it into the air. We're going to talk about droplet nuclei floating through the air. You're going to inhale it, it's going to stay in the upper respiratory system, it's going to cause pharyngitis or sore throat. Then it's going to cause tonsillitis, and then the sequelae, if it has a specific super antigen, we're going to talk about that just now. Not every strain of strep pyogenes has the ability of making a super antigen. The super antigen is only found in strains that are associated with something called scarlet fever, and we'll talk about that. Okay? So we're going to talk about this. On the left, though, is all the possibility complications that can go through. So at the bottom, what we're worried about is glomerulonephritis and rheumatic fever. You guys have already heard about rheumatic fever once before because we talked about it with subacute bacterial endocarditis. We'll talk about why that's important and why it's associated now. So let's talk about this. So let's go over what a strep sore throat is. First of all, a strep sore throat, I have to show you good pictures because this is where I get to gross you guys out completely and always waste you guys out. Okay? So let's lower the light, snuggle in, and let's look at our favorite diseases. So, strep sore throat, we have a local inflammation. It's a local inflammation and a localized fever. It's not going to go systemic. Okay? You're going to have, so there will be a fever sometimes, but again, it can be a low-grade fever, it can be a mild fever, it can be just a sore throat. Then we're going to have pharyngitis and tonsillitis, and you guys all know what tonsillitis is when your tonsils swell up. It's an inflammation of the tonsils. So tonsils are just lymph nodes at the back of your throat. When you see this figure from the book, this is not very, I don't think, illustrative of a strep sore throat because they talk about red patches with white dots, patches on the back. That is pus. That is PMNs being called. That is the infection trying to be eliminated by our immune system. So let's look at some other figures from again, you have red throat with white patches and a fever, generally don't have other symptoms, no runny nose, no weepy eyes. So this is another figure from the book about what strep sore throat looks like, group A disease. So when we look at this is white patches on the back of the tonsils. Oh my goodness, look at these white patches on the back of the tonsils back here. Yeah, that's all pus, that's bacteria, that's the immune system fighting off this infection. So this is all your enlarged tonsils. This is that thing that dangles down in the back of your throat. You guys know that from physiology, and it begins with U. I don't remember what it is. A uvula, something like that. Okay, so that's what it looks like. Now, the other thing that happens is students get so excited about this class that when they get the infection, they send me their own pictures. So one of my students said, Dr. Kramer, I had strep sore throat. I had to show you what it looked like. And this is an actual physical picture of a student's strep sore throat. Okay? And then when I showed that, somebody decided they tried to outdo her. And then this was their other strep sore throat. So when we say it's tonsillitis with white patches and red, it's an infection and an inflammation. There's pus patches on the tonsils, on the back of the throat. Okay? One of the things that's important is that we now have something called an indirect agglutination test. So we're going to agglutinate. What they will do is they take a swab of the back of your throat. And they will, if there are bacteria there, they will agglutinate with antibodies to these bacteria. If it agglutinates, you have a strep sore throat. If it doesn't agglutinate, most likely what you have is a virus. And they won't give you antibiotics for it because it's a virus. So, the other thing that they will tell you, and what they should tell you, is that if it's a negative test, sometimes it's a false negative, you might still have strep sore throat, they will do an incubation test where they will swab a plate and look for strep pyogenes on a plate after 24 hours. And then they will call you to confirm either that you don't have strep or that you do have strep. And if you do have strep, then is when you will go and get the prescription filled that they gave you. You do not start the prescription until they confirm that it's a bacterial infection because there are very many causes mimicking a strep sore throat. Does that make sense? Because if it's a virus, antibiotics won't help you. Okay? So, there's a 10-minute agglutination test that will have to be confirmed by culturing in a lab and identifying strep pyogenes just like you guys are going to do in a lab or like you did in a lab. Now, if the strain of strep has that super antigen, if it has a super antigen, then it can make additional signs and symptoms of a strep sore throat. Now, commonly, this super antigen doesn't cause much more serious sequelae than a normal strep sore throat. So when we talk about scarlet fever is due to a super antigen. You will get a rash, and you will get this is an erythrogenic toxin. So you can see the rash on the child's skin right here. This is the rash due to the superantigen activating T helper cells nonspecifically, but it's not a toxic activation. So we'll get a rash, and we will get something called strawberry tongue. They call it strawberry tongue because your tongue looks like a strawberry. This looks like the top of the strawberry, right? So all of that is due to the superantigen production. It is just a sore throat. It is just and will be. But if you have scarlet fever, you know by definition that this is strep pyogenes, right? So if you just have the regular sore throat with splotches on the back of your throat, it may or may not be strep throat. Scarlet fever is strep sore throat, okay? Because it is one of the toxins that is secreted. This toxin is secreted into the bloodstream. It becomes toxemia. It targets T helper cells, non-specifically being activated by antigen-presenting cells, and we'll get this small cytokine flash, okay? It's normally not a problem because we will give you antibiotics just like we would have if you'd had a non-scarlet fever strep sore throat. As a matter of fact, there is a huge outbreak of scarlet fever in Europe right now, especially in the UK. There's been this ongoing scarlet fever outbreak but the thing is all they have to do is give antibiotics. And in this case, because it's a gram-positive cocci, for some reason, strep pyogenes is still exquisitely sensitive to things like amoxicillin and even penicillin. So they will give you amoxicillin for strep sore throat or penicillin. All of you guys who've had a strep sore throat know this. You've gotten amoxicillin prescriptions, right? Because this is still capable of acting and destroying these bacteria. Now, what we worry about, again, this is the superantigen. It binds outside the MHC class II molecule, and then it activates nonspecifically this T helper cell because this T helper cell would normally not recognize this antigen because it's not the same shape. It doesn't recognize the MHC molecule, but the superantigen is inducing that activation. So let's talk about what we are worried about. What we are worried about is acute rheumatic fever. And acute rheumatic fever is a type 2 hypersensitivity. And the reason that makes a difference here is because of molecular mimicry. Because some people have a specific MHC molecule, they present a specific peptide, they activate an antibody response to a specific M protein. These antibodies then cross-react not just with the M proteins. Not all M proteins do this. Not all MHC molecules do this. Certain strains and certain MHC molecules allow the production of these autoreactive antibodies. And these autoreactive antibodies bind to the mitral valve of the heart, where they cause scarring. And as a matter of fact, people who've had rheumatic fever can have that heart murmur because the valve doesn't work as well anymore. It doesn't flop the way it's supposed to because it's scarred. So there's blood that leaks through because it's not very good at stopping the blood because it's scarred and it doesn't work as well because we've made an acquired immune response to this organism that cross-reacts with self. So it is a type 2 hypersensitivity because antibodies are binding to self-tissue. We will activate PMNs, we will activate tissue degradation, we will have all of that happen. And once that scarring happens, this is when subacute bacterial endocarditis comes in. This is when, if you've had rheumatic fever, that scarring allows for your normal microbiota to have a place to bind, to hold on to, and to cause a biofilm to form. And when that happens, we get embolisms, we can get clotting, we can get strokes, we can have all of this. Okay? So again, don't confuse scarlet fever, which is just a normal strep sore throat, with acute rheumatic fever. There's fever in both names. Okay? So acute rheumatic fever is a hypersensitivity response following the infection. So this is why we want to give antibiotics as early as we do, because the earlier we give you antibiotics, the less chance that we have of producing an antibody response, particularly an IgG response, that allows you to make an IgG response to these M proteins. So if we can stop them with antibiotics, we stop the acquired immune response from kicking in. Okay? So that is what we want to do. That is one of the sequelae of having a strep sore throat. This is why it's important. This is why when I tell you not to take antibiotics, I tell you not to take antibiotics for non-important things, for any viral infections. This is one of the diseases where you have to take antibiotics. And you have to go in and catch it as early as you can. And you have to be on target for this. Now, this is more important. As we get older, we make less and less of a problematic response. Like, I never go in and worry about it. If I have a sore throat, I don't worry about it being a strep sore throat anymore because my immune system, I think it just becomes tolerized to strep. Or I've had enough of a response that it's not going to cause a response. But when children get this infection, you specifically need to make sure you go in and get it tested so that we know if it's a strep sore throat or not. And if it's a strep sore throat, it's absolutely vital that you give your child antibiotics. Okay? Antibiotics are a miracle drug. Antibiotics are what keeping us from having those terrible heart-rending books where the little kid is going to die from rheumatic fever. Antibiotics will save people. This is one of the diseases you need to use antibiotics for. Okay? So, this is one of the sequelae of a strep sore throat. So let's talk about another sequelae of a strep sore throat. Again, the other thing that you need to understand is that we talked about how EBV is the leading cause of cancer in third world countries. Now, this is the leading cause of heart disease in third world countries, and the reason is that in third world countries, you might be 15 miles by foot from your nearest doctor who can give you antibiotics. So your kid isn't going to get antibiotics for the sore throat. So when we talk about disease in third world countries where they're not as capable of having appropriate health care, appropriate timely health care, they're going to get heart disease because they're going to make antibodies to this protein, this M proteins, and they will cross-react with their mitral valves and have heart disease. We have helicopter parents. You can't tell me that all of you, as soon as you saw the first signs of a fever and again, there is a fever with this, first signs of a fever and a sore throat, that your mother didn't whisk you into the doctor as quickly as possible, demanding antibiotics. I know they did. Okay? So, this is one of the reasons we don't have as much rheumatic fever in the United States. Okay? So the other thing that we're going to talk about is the second sequelae, and the second sequelae is, I did back up from that fever thing. There's another disease. Strep sore throat doesn't have a fever associated with it. Some people don't have a fever with it, but that's not as common. Glomerulonephritis, we've already talked about glomerulonephritis, and you guys already know that glomerulonephritis is a type 3 hypersensitivity. This is also antibodies to those, these are not the M proteins, but these are antibodies to the bacterial infection itself, to surface proteins on the bacteria. These cause small antigen antibody complexes that then get filtered out into the glomeruli, where they activate complement, they activate PMNs, and it causes the destruction of the kidneys. When the kidneys are destroyed, you'll never get a kidney back. The kidneys are gone. You will need a kidney transplant if this happens to both kidneys. One functioning kidney is okay, but if both kidneys are destroyed, you'll have to have a kidney transplant, or your kid will be on dialysis for the rest of their life. So, what we see, and one of the ways that we can determine if you're having glomerulonephritis, is there'll be more protein and blood in the urine because your kidney, your glomeruli have been destroyed, just like your capillaries would have been destroyed in DIC. So it's going to allow more blood and more protein to get into the urine, and that would be indicative of glomerulonephritis. So those are the two real reasons that we want to stop a strep sore throat. Not because it causes a sore throat, not because the sore throat is dangerous. It's our immune response to the sore throat that we worry about. So let's go back over this. We're going to have a red sore throat, pus, hemorrhages, enlarging enlargement of the lymph nodes, particularly the lymph nodes, the tonsils, which are lymph nodes in the back of your throat. So you could have abscess formation, and that makes sense from seeing those pictures. goodness, if there's enough of those white patches, you can eat away at the tissue until you get blood into the bloodstream. Involving tonsils, occasionally rheumatic fever and glomerulonephritis as sequelae. Okay? So it's strep pyogenes, Lancefield group A, beta hemolytic strep, associated with hyaluronic acid capsule and M protein, both of which inhibit phagocytosis. Okay? Protein G binds the Fc segment of IgG, protein F for mucosal attachment, multiple enzymes. So the enzymes depend, if they have the superantigen toxin, then they will make scarlet fever. Okay? And then off to the left, you see rheumatic fever, glomerulonephritis. We're not going to talk about neurological abnormalities or complications, but damaged heart valves leak and heart failure can develop as well if enough scarring occurs. Question?
Is there a specific reason why rheumatic fever is back to elbows and knees as well?
Oh, I don't know about elbows and knees. 5. Yeah, I don't know. I don't know. Because it's associated with the heart. I don't know. Maybe there's joint problems too, but that's a good question. I mean, nobody's ever noticed that before. Joint pain. Okay, thank you. I didn't know that. Okay. Good job. Okay. So now we're going to talk about diphtheria. Diphtheria is a dread disease. Diphtheria kills. Diphtheria can be spread, and we're seeing an unnatural, an unwanted, and unnecessary rise in cases of diphtheria worldwide. We'll talk about why in a minute. It causes a sore throat. The first thing that happens is you're going to get a sore throat and a fever. And then you'll also have fatigue and malaise. When you have fatigue and malaise, that's a prodromal stage. The other thing that's going to happen is we're going to talk about the rest of the signs and symptoms now. I'm not going to ask you about the incubation stage. The organism that causes this disease is Corynebacteria diphtheriae. Now, what you do need to know is you need to be able to identify the causative organism of diphtheria. There's a big hint in the name. Corynebacteria diphtheriae. As long as you can recognize it, you don't have to spell it. I have to spell it. And sometimes I even mess that up. Okay? You just need to identify it. You also need to know that it is one of the few gram-positive rods we will be discussing. It is also the virulence factor that makes this a disease that kills is an AB toxin that is caused by something called lysogenic conversion. Have you guys heard of lysogenic conversion before? Thank you. For those who nodded a little bit, kind of like, oh, yeah, yeah. Okay, so let's talk about this. We're going to talk about how Corynebacteria is spread. It's through respiratory droplets. So it's just spread through respiratory droplets. And it's going to first infect, and it only infects the upper respiratory, the throat. So you're going to have a sore throat. It's going to infect the ciliate epithelia of the respiratory system. One of the things, once it binds to the ciliated epithelial cells, it is going to start secreting a toxin, and it's going to grow. And where it grows and secretes this toxin, it's going to cause an inflammatory response. And not only will it cause an inflammatory response, but it causes this thing called the pseudomembrane. And it's called a pseudomembrane because it has that, it looks like pus. It kind of makes, the reason we call it a membrane, because it's not pus. It's not something that you can wipe off with a Q-tip. In order to remove this pseudomembrane, you have to remove it with a scalpel. It is fibrous. It has to be cut away. And if it continues to grow, it can actually block the breathing tube and suffocate individuals if it's allowed to continue. So this organism only grows on the ciliated epithelial cells. It does not become bacteremic. It does not do anything other than start secreting its AB toxin. And its AB toxin is called diphtheria toxin. How about that for being nice? Okay, so we'll talk about this. So when the organism is growing, the first thing that we're going to see is a pseudomembrane, a local effect. And then the other thing that's going to happen is that is going to start then secreting the toxin into the bloodstream, and we're going to have toxemia. The B portion of that AB toxin will then bind to its target tissue. It's not all tissue, but the tissue it binds to is kind of important. Let's look at what it binds to. It binds to the heart, it binds to the nerves, and it binds to the kidneys. So when it binds to these tissues, it only infects those tissues, and when it gets bound by the B fragment, it causes those tissues, those cells, to take that AB toxin in, and when it's taken in, you guys know that acidity causes the A portion to be cleaved from the B portion and activates that enzyme. So it's not toxic in the bloodstream. It's not toxic when it's circulating in your body. It's only toxic when it enters these tissues. And then when it enters these tissues, it's going to kill the tissue it has infected. This is because the AB toxin, the B portion binds, the A portion is activated, and then we're going to activate the enzyme. And when we activate the enzyme in diphtheria, again, this is caused by a lysogenic conversion. This is a lysogenic phage that's infected these organisms, has integrated its chromosome into these organisms, and the actual diphtheria toxin is encoded by the bacteriophage genome. Now, almost all Corynebacterium diphtheria have this phage. Almost all it produces this toxin. It allows it to outcompete other organisms that don't have this phage. So, what else do we know about this organism? What else do we know about this toxin? This toxin, this is one of the first most dread diseases, and this is one of the first ever vaccines. And the vaccine, the homework quiz for vaccines in Chapter 19 will be available tomorrow at noon, maybe a little afternoon, depending on if I'm running a little late. Don't email me if you try to get on exactly at noon and it's not up yet. I promise it will be coming. However, for this vaccine, we are going to make an AB. We're going to make a diphtheria toxoid, meaning we're going to take that AB toxin, we're going to modify it so it can't be cleaved, but the antibody response will still neutralize the B portion and not allow it to bind to its target. If that toxin can't bind to its target, it is just a protein floating in your blood. If it can't bind to its target, it can't have an effect. So if we have neutralizing antibodies present, when you get infected with this organism, you won't have disease because disease is only caused by the toxin. You might have a small sore throat, but nothing else. So let's talk about how the toxin works. The other thing is the toxin, the toxoid, is part of a vaccine. You may have heard of it. We call it the DPT vaccine. It used to be the DPT vaccine, and then it became the DTAP vaccine. There's different formulations of it. Some are for children, some are for adults. I'm not going to ask you which one is for children and which one are for adults. But the D stands for the diphtheria toxoid. So this is one of 3 organisms that is contained in this vaccine. So we need a booster of it every 10 years because or every so often for the diphtheria is because this toxoid is just a protein. It's not an inactivated or not attenuated organism. So when we inject it into your body sees it as something that's different or not self. We will make a small antibody response to it. We give it to you a number of times to increase it so that you have a neutralizing antibody response. But you have to have this updated all the time. Otherwise, you won't have a neutralizing antibody response. And the most important thing that you need to understand is just how lethal this toxin is. This toxin only needs very, very, very small amounts to be produced in order to kill heart tissue, in order to kill a kidney, in order to cause neurodegenerative problems. Small amounts. So when your child is having a sore throat, you might not realize that that is an organism that is also secreting a toxin that's targeting their heart. Okay? So, We are going to talk about Corynebacterium is everywhere. It's in respiratory tract. People carry it. Older individuals who've had this before or who've been vaccinated can get a milder form of the disease because they already have an existing immunity to it, and so they will fight off the infection more rapidly. However, if I am holding a newborn or child under the age of 1 and I'm breathing respiratory droplets with Corynebacteria diphtheria on it, I will be infecting that child. There's now a commercial out where the grandmother becomes the big bad wolf as she's breathing onto her newborn grandchild. So we can have diphtheria without the classic signs and the symptoms because we have an immune response if we've been vaccinated at all. We'll have a milder form of the disease because it's just the toxin that kills us. And because we've had the vaccine, the toxin will activate the memory B cells that were made back when I got the vaccine to cause a more neutralizing antibody response to make sure that I'm protected. But children who don't get vaccinated are not protected. And when they're not protected, it's small amounts of toxin that will kill their heart, destroy their kidneys, and they cause all sorts of problems. Paralysis. Because you haven't vaccinated your child because you're worried about the vaccine causing other sequelae. It has been proven over and over again that the DPT vaccine does not cause autism. They have proven it in a number of ways. They have proven that in some nations, like in Sweden, they have to track everybody. And they actually proved that autism went up in the very small, not a statistically significant amount, but autism rates went up in unvaccinated children. This is not a vaccine that causes sequelae. Now we're beginning to understand that it might be stupid things like the stuff we're spraying on your oranges that does it. But we're not going to vaccinate our child. We're going to let our child eat organisms or fruit that has stuff on it. So, or just so you know, most of the chemicals that we make to put on our grass to make our lawns green, because the EPA has been told not to worry about this, they're not tested on its effect on humans or children. So all of those chemicals you put on your lawn to make your lawns green and to control the insects on that makes your lawns green and kills your children, or maybe gives them autism. So, this is what I'm saying. This is a dread disease. It killed thousands of kids before we had this vaccine. It caused all sorts of problems. And the reason why people think that it's not a problem anymore is because they were vaccinated by their parents and their parents had been vaccinated. So we didn't see the side effects of this infection much until people stopped vaccinating. Okay, so we're going to talk about this throughout the rest of the class. This is the DPT vaccine. The toxin is released. The pseudomembrane forms. Toxin can cause paralysis, damage to the heart muscle, kidneys, and nerves. Infection in the upper respiratory tract. Exotoxin is released. Have receptors, mainly heart, kidney, and nervous tissue. I'm not going to ask you what to treat with, but this is a bacterial infection. It doesn't matter if you start an antibiotic therapy. Once they have the infection, if they have never been vaccinated before, giving antibiotics when you notice signs and symptoms is too late. Damage of some sort has occurred because small amounts of this toxin are toxic. Okay? Questions about this? We are seeing huge increases, spikes worldwide because people have stopped taking vaccines. And it's all due getting vaccines. And it's all due because one person wanted to sell their different vaccine and utilized again, we're going to talk about experimental procedures in chapter 19. He picked 12 children that had symptoms. These were not randomized. These were not anything. He picked 12 children who had already had symptoms and proved what he wanted to prove. His work was discredited. You don't get disbarred from the medical profession. You get un-MD'd. He was un-MD'd. What is that called? He had his stethoscope ripped off of him, okay? He keeps fighting back, but nobody, all of the data that he proved, that he published, was withdrawn and retracted. And all of the people who've tried to find anything, we have tried to prove over and over and over again that any of this could be true. And it has never been reproduced or been able to be reproduced by anyone. Okay? We're going to talk about this. As a matter of fact, I'm going to link something on my blog. I'm going to start you guys looking at my blog because I want more hits on my blog, on all seriousness. So as a matter of fact, on my blog, I just updated a post about yellow fever. The day that we talked about yellow fever, I just got an inbox alert that there was an outbreak of yellow fever from December of last year through March, the end of March of this year. There were over 1,000 cases of yellow fever in South America with a 33% death rate. And there's a vaccine available, but it's not available to the very poor. And there are some places where it's not available because it's just not available. And yet these countries where yellow fever is endemic don't make sure that all of their people get vaccinated. It has a 33% death rate. Okay, it's a vaccine. If you knew something caused a 33% death rate, would you get vaccinated? Absolutely. If there was a Zika vaccine, would you get the Zika vaccine? Absolutely. So we're going to talk about this as we go. We get more and more vaccines because we have the ability to control these infections because we've learned how to stop the infection.
Is Zika like a big concern in the U.S. like right now?
Is Zika a big concern in the U.S. right now? I have to tell you, we have a whole part of our virology department they're all focusing on Zika right now. Just because even if it's not a problem right now, it may be a problem because it can be sexually spread. People can go there. We'll talk about Zika later, okay? So, because Zika is not respiratory spread. So, this is diphtheria. Prevention by immunization, toxoid, given to children at 6 weeks, 4 months, 6 months, 18 months. And even when we give them, their immune system isn't as good as an adult's immune system. So, their immune system still doesn't protect them as well as our immune systems protect us. And for the children who don't get vaccinated, we're going to talk about herd immunity in chapter 19. It is up to us. I get boosters because I don't want my sore throat to be diphtheria and me to be a vector for disease. Okay? Okay. We're going to talk about the common cold. I love the common cold. It is one of my favorite diseases. Okay? Just so you know, in our department, I tell you about DNA viruses and RNA viruses, and there's whole meetings for this. In our Virology in Progress seminar that we have every Friday, they actually had a family feud between DNA virologists and RNA virologists. Yes. And I have to tell you, the DNA virologists cheated by having only interviewed their people. So the RNA virologists didn't know any of the answers because they don't know. They don't follow RNA viruses. This is an RNA virus. That's why it's important. All of the organisms that cause this disease are generally RNA viruses. We think. We don't know. The reason that we're talking about this is that you guys all know what the common cold feels like. You're going to have a scratchy throat, nasal discharge, malaise. You're going to say, if I give you malaise, malaise is that crappy feeling that you feel like you know something's happening and you're coming down with something. That's malaise, okay? A headache and a cough. Now, I am going to point out the incubation period. You don't need to know the incubation period, but I do want you to see that the incubation period is 1 to 2 days. So if your friend has a cold and all of a sudden you get malaise, you can point to the person who gave you the cold, most likely. Now, we'll talk about this, differences between this. So we have the cold. The organisms that cause the cold are mainly, we talk about rhinoviruses. Rhinoviruses are an RNA, a naked RNA virus. It is a naked RNA virus. Because it's a naked RNA virus, it can't be controlled by alcohol hand sanitizers very well. because it's a naked virus. And you guys already know that things like Lysol and things like those common disinfectants won't control a naked virus. They don't talk about that on the packaging when they talk about being able to control infectious organisms. 99.9% of all infectious organisms that are controlled are the ones on the back of the page, and rhinoviruses aren't one of those. Okay? Because it's a naked RNA virus. The other thing that happens with this naked RNA virus are that there are 100 different strains or types of virus. Okay, so there are 100 different strains or types. That means that you can have 100 different infections with this organism and never have the same virus twice. And when you don't have the same virus twice, that means that you can't make an immune system to a different virus because you haven't seen it yet. The other thing that's really interesting is, remember, it's an RNA virus. So it has an RDRP. And when we talked about antigenic variation, we talked in chapter 16 about how RNA viruses, RDRPs, are RNA-dependent, RNA polymerase, and RNA-dependent RNA polymerase. They don't have the proofreading ability that DNA polymerases do. So these viruses drift antigenically. They drift, meaning that they will change their antigens. And so the virus that you got last year might have drifted, so even though there are 99 more strains out there, you're going to be reinfected with the exact freaking virus that you had, not the exact freaking virus that you had last year, but one that had migrated a little bit antigenically so that your antibodies no longer bind to it. The other thing is that the toll-like receptors and the RLRs and the NLRs that recognize this is not a very serious or dread disease. So we don't make a very strong response. We make a mild response. And so as a matter of fact, when I told you that we can find antibodies to your infectious organisms decades later, it's not true for the cold. Your immune system forgets these organisms over time. So not only will you have had 100 infections from the 100 different strains, and then multiply that by every time they change antigenically, and then multiply that because you won't have any long-lasting protective immunity because we just forget what it looked like. It's kind of like one of those science fiction fantasies where the man you can't remember what his face looks like because you can't recognize me. So we just don't recognize it again. So we get it over and over again. So the problem is how easy is it going to make a vaccine to something that drifts antigenically, that we don't know where it's going to go, how it's going to drift. And then on top of that, if you read the book, that this only causes about, all those 100 strains of rhinoviruses only cause about 50% of all known colds. There's something called a coronavirus, that's an RNA virus that can drift antigenically, that causes about 30%. And then about 20%, you guys don't need to know these percentages. Just know that there are other organisms that cause the cold. 20% of all organisms that cause the common cold, we don't know what they are. So how easy is it going to be for us to make a vaccine to something that drifts antigenically, has 100 different strains, and 20% of which we don't know what it is? Yeah, that's not going to happen. So you're going to get the cold. And you're going to have, they attach to your respiratory epithelium. And one of the problems is that when they attach to your epithelium, it's going to spread to adjacent cells, and your ciliary action is going to cease. We're going to see this happen again and again because the cells that they infect are your ciliated epithelial cells. And when your ciliated epithelial cells die, they don't move mucus. And when they don't move mucus, the only way that you can move mucus is through something called coughing. So you're going to start coughing and sneezing in order to move the mucus from your lungs. And so the more you cough and sneeze, and the thing is this coughing and sneezing can go on until you regrow your ciliated epithelial cells. Does anybody know how long this coughing can go on normally? So the cough itself, the infection itself can last about 8 days. You can keep coughing for up to 18 days after you have this infection because your ciliated epithelial cells have to grow back. So people are saying, I don't know, I'm not getting over this. I still have this terrible cough. Well, it's because you don't have ciliated epithelium. Now, because this is an acute infection, you're going to eliminate this organism from your body entirely, and there's no latency there. But you will have also had your ciliated epithelial cells die. Mucus secretion increases because it wants to get rid of these organisms. So you're going to have snot and mucus, and then you're going to cough and wheeze, and then you're going to have all that happen. You guys know exactly what happens. Infection is stopped by interferon release. Is that interferon alpha beta or interferon gamma? Interferon alpha beta, because this is a virus. Okay? So interferon alpha beta helps stop this infection. It induces inactive antiviral proteins. When the virus gets into the new ciliated epithelial cells, it activates those inactive antiviral proteins to become activated antiviral proteins, and it will kill the ciliated epithelial cells, which spreads the ability of cell death. More mucus, more coughing, more watery eyes. Okay? Also, notice that it says an ID 50 of 1. Now, portal of entry is important. We're going to talk about portal of entry in chapter 19. Portal of entry is there was a, this is where we get no fever. I'm sorry. When we have a common cold, you generally don't have a fever. When you have a strep sore throat, you have a fever. When you have a common cold, you normally don't have a fever. So the two signs and symptoms are completely different. So with a strep sore throat, you will have a fever, but no watery eyes, no runny nose. With a cold, you will have no fever, but watery eyes, runny nose, cough, drip, all of that. Okay? So when we talk about this, there's usually no fever. It's a naked RNA virus. Those alcohol wipes don't work. Or those hand sanitizers won't work. And again, there's no generally accepted treatment except for hand washing. If you wash your hands, you prevent transfer of disease. The other thing is portal of entry is important. They did studies back in the 80s, where they looked at how rhinoviruses spread. And so they put people, there were 3 studies done, and they put people who had the cold in one side of the room, and they put people who didn't have the cold in the other side of the room. They paid these individuals who had colds to come in and sneeze in the presence of people who didn't have colds. It gets better, wait. Then they looked, and only about 13 to about 18%, not even a fifth of the people came down with the cold. So it's not really spread really well by respiratory droplets. So then they thought maybe it's spread by something called a fomite, or innate things. So they innate things like a water bottle, or like playing cards. So they soaked a deck of playing cards in nasal secretions from somebody who had a cold, and then they had people play with these cards that were soaked in the virus. And only about 18% of those people came down with a cold. And then the third study that was done was they had a person with a cold kiss a person without a cold. Now again, these people didn't know each other. Okay? The person with the cold, come here, and they had a kiss for 60 seconds. And still, it was only about 18% infectious rate. But you know what will get you every time? Is when you rub your eye with your finger that has a virus on it. One virus, one portal, allows that to drain into your sinus cavity. It inoculates the tissue that is infectious with this virus, and it gets in and it'll get you every time. So washing your hands to keep the virus off is important, and not touching your face is important. See how long you can stop touching your face. Stop touching your face. Don't rub your eyes. Don't touch your face. See how long you can do that. I personally think that when I get a virus on my finger, it somehow transmits nervous impulses into my arm to want me to bring it to my eye, because I find that when I wash my hands, I want to rub my eyes a lot less. When I'm on the bus, I suddenly want to rub my eyes all the time. Okay? That is a hypothesis I will prove sometime in the near future. Okay? So there's no acceptable treatment. It's a virus. Once you get the cold, the only thing that you're going to do is you're going to take anti-cold medicine. And the anti-cold medicine reduces mucus, reduces coughing, reduces sneezing, reduces watery eyes, and reduces all of the signs and symptoms. And what you're reducing is your innate immune response. You feel better, So yes, I take anti-cold medicine if I have to come into work. I've tried to muddle through. It's a pain in the butt to muddle through without cold medicine. But if you take cold medicine, they've done 2 studies. Would it reduce transmission because you're not sneezing as much? Well, what they found is it does reduce transmission because you're not sneezing as much, but because you shed the virus for much, much longer, you are more infectious for longer periods of time because you've stopped your innate immune response. How cruel is that? Science and facts work, okay?
Probably a stupid question.
There are no stupid questions.
Is it called cold because of the absence of fever?
I don't know. That's a good question. I don't know why it's called a cold. Somebody find out. If you find out, let me know. Okay? You are shaken in cold, yes. Okay, so let's look at the next one. Okay, transmitted, we don't know. RNA virus mutates, antigenic drift, and then no vaccine, hand washing. Antibiotics will not work. As a matter of fact, they have come up with a cold medicine. So the normal period of time for a cold is about 8 days. Because again, it doesn't take much of an acquired immune response. We'll make some IgA, but non-long-lasting IgA. We'll make interferon alpha and beta, but that goes away. So one of the things that happens is people go into the doctors and they will demand antibiotics for their cold, even though they know it's a cold. And they go in and demand antibiotics for a cold, and the doctor will say, you have a cold. And they're like, I know, I had this last year. I took antibiotics and it helped. So they go in on day 2 or day 3 when they're feeling bad. They get a prescription on day 2 or day 3 when they're feeling bad. And lo and behold, in 2 days they start feeling better. And by 5 days it's over. Which would have happened even if they hadn't taken antibiotics. Because it only lasts about 8 days. Their cough will still remain. Antibiotics won't help that. Antibiotics won't work on a cold. All of the organisms that cause a common cold are all viruses. Okay? Well, okay, asterisk. Except those 20% that we don't know what they are and what they could be. Who knows? It could be aliens from outer space that are causing the other 20%. Okay? So, antibiotics wouldn't work on them either. Okay? So, we do have some medication that doctors, if you're going to be a doctor or a PA, you can give this to your patient instead of antibiotics. It will reduce the signs and symptoms of a cold from 8 days to 6. So you're going to pay for this anti-cold medicine that will reduce the signs and symptoms from 8 days until 6. It won't cure you. It won't stop the infection. It won't make you less infectious. But your signs and symptoms, your infection will go away more rapidly. Okay? So now, let's talk about adenoviral pharyngitis. Adenoviral pharyngitis, you can kind of guess that this is an adenovirus. And as a matter of fact, we are certain that there was an adenovirus going through the university and through the campus last fall, about November and December. It caused a horrific long-term infection, meaning it felt like the flu, but it wasn't. So you had cold symptoms, and as a matter of fact, there are 45 different strains of adenovirus. Each strain causes a different severity of disease. Some of them cause conjunctivitis. Some of them cause a severe enough infection that it's like, it can cause pneumonia because it's such a severe upper respiratory infection, depending on the strain. I had it and I was down for 3 days where I did not want to move. And it was just a coronavirus. So this is the kind of virus, adenoviruses. This is a DNA virus. So this is also a DNA virus that doesn't cause, we don't think it causes, this is one of the DNA viruses that doesn't cause cancer. However, the different severity, all of them will cause a fever, a very sore throat, a severe cough, swollen lymph nodes of the neck, and pus on the tonsils and the throat. So when you look at those signs and symptoms, what does that look like? A strep sore throat. This is the organism that mimics a strep sore throat. This is why we have the rapid strep test. This is because if you have something that looks like a strep sore throat and smells like a strep sore throat, but there's no strep present, won't be caused by strep pyogenes. Probably caused by a coronavirus, this adenovirus. I'm sorry, it's an adenovirus. It's not a coronavirus. It's an adenovirus. It's a DNA virus. The virus multiplies in the host cell, destroys the cells, causes inflammation, and different types produce different symptoms. Some coronaviruses cause conjunctivitis. It makes your eyes turn really red and swollen. Okay? This is what, so I had to go online and look for a picture of what adenoviral pharyngitis looked like. Tell me that you can differentiate adenoviral pharyngitis from a strep sore throat. This is why we have the test. This is why we check. One is a virus that can't be treated with antibiotics. One is a bacteria that has to be treated with antibiotics. Are we good? This is why I include adenovirus in my talk every year. People say, well, it's not that bad. No, it's not that bad. It's not going to cause sequelae. It doesn't have M proteins. It's not going to cause rheumatic fever. It's not going to cause anything. It's just going to cause a sore throat, and we call it a self-limiting sore throat, meaning you will get over it. I felt crappy for 3 weeks, 3 long agonizing weeks, and I felt like I was never going to get over it. And everybody I know that had this went in and got antibiotics for it, except for me. Yay, me. Everybody give me a little hand. Okay, because it was hard. It was hard and unpleasant. But I knew it was a virus, and there's nothing I could do. So I ate chicken soup, I got a lot of rest, and I caught up on Netflix. Okay? How did I know? I knew the signs and symptoms, and I knew it wasn't the flu. So I'd gotten the flu vaccine, and I know what the flu is. So yeah, it was just upper respiratory. There was nothing else with it. So it was an upper respiratory infection. It was much more severe. And just because you know these, it's much more severe. It was a severe upper respiratory infection, and it wasn't strep sore throat. Question?
How did you know it wasn't strep sore throat?
Because I didn't have any of the other classic signs of a strep sore throat. And a strep sore throat doesn't last 3 weeks. Okay? And because I was feeling like, I could tell it was interferon alpha and beta. Interferon alpha and beta just makes you feel crappy, okay? So, this is adenovirus. So the other thing that I want, again, there's no treatment for symptoms. There's no treatment except for relief of symptoms. There's nothing you can do once you get an adenovirus sore throat. There's nothing you can do but muddle through and baby yourself as much as you can and baby your immune system so that it quickly and efficiently activates your T cytotoxic cells. One of the things we know is that it takes a lot of energy to generate all of those clones. So you need to keep yourself on track. You need to keep yourself nourished. You need to keep yourself hydrated, and you need to keep yourself from walking around and infecting other people. There's no vaccine. Wash your hands and avoid people with symptoms. And we'll finish this up on Thursday. Or what's today? Today is Thursday. What's today? Thursday. Okay.